Hello students, welcome back to the second session of my lecture and welcome back after the break. You are listening to Sandeepan Sengupta, an assistant professor in microbiology at the Kingston College of Science. So we were studying and discussing about the two different forms of mutagenic physical radiations or physical mutagens, that is ionizing radiations and non-ionizing radiations. So let us recapitulate a little bit of that. So what do we mean by ionizing radiations? Those radiations which actually hit the biomolecules and cause them to lose electrons and hence ionize them in course of the process are ionizing radiations. These are the radiations of very short wavelength which have very high energy and are enriched with a high source of energy. Ionizing radiations, the two major forms of ionizing radiations which are known to generate mutations are gamma rays and X-rays. And what do you mean by non-ionizing radiation? Non-ionizing radiation actually refers to ultraviolet radiation, being a bit higher in wavelength, that is, and a bit lower in energy. Ultraviolet, that is UV light, is known to affect the macromolecules in a cell, hit a cell and affect the macromolecules, proteins and DNA in a cell by generating in a different way, which does not involve ionization of these macromolecules, but rather involves a process called dimerization of the nitrogen bases present in DNA, that is of nucleotides, nitrogen bases of nucleotides. So this is the major difference between the ionizing and non-ionizing radiations. Now, what are the different sources of ionizing radiations? There are few more differences. Ionizing radiations are of higher energy and are of lesser wavelength, while non-ionizing radiations, so-called the UV or non-ionizing form of physical mutagen, that is ultraviolet radiation, is of higher wavelength and lesser energy. And because of higher energy, ionizing radiation is able to penetrate well through all opaque surfaces, through glass surfaces, and is easily accessible to all kind of gametes during the course of the replication cycle in order to generate mutation. But UV ray, being non-ionizing in nature, is not able to cause ionization of DNA and proteins. This is because of the fact that they are less in energy. Hence, the UV rays or the ultraviolet rays, being lesser in energy and being of higher wavelength, and are not able to penetrate or pass through opaque objects than certain glass materials, and hence they can generate or affect the biomolecules, that is proteins and DNA, of only exposed cells present exposed at surfaces. They can only act over exposed cells. Now, the third major difference is that one more difference is that there lies in the source of the ionizing and non-ionizing radiations. The two forms of ionizing radiations which act as physical mutagens are X-rays and gamma rays. So the important sources of this are X-ray machines, then cathode ray tubes, then certain radioactive decay of nuclides or radioactive decay of nucleotide, that is nuclides, or radioactive decay of substances like cobalt-60, then cesium-127. Of course, these are the major sources which generate ionizing radiations. X-ray machines generate X-rays, then gamma rays are being produced as a result of decay of radioactive nuclei like cobalt-60, then cesium-127, etc. And what is the source of non-ionizing radiation? Actually, the visible light, is the source of non-ionizing radiation. The light is the source of non-ionizing radiation, that is UV radiation, or the prominent physical non-ionizing physical mutagen. Now, or the most important source which is applied of non-ionizing radiation or UV radiation, which is used in genetics, is a UV lamp or a germicidal lamp. Now, germicidal lamp emits UV radiation, and UV radiation does not ionize the proteins and DNA, but rather produces an effect in DNA in a different manner. Now, let us peep into before directly moving on to the mechanism of these radiations, let us discuss a bit about the way the radiations are measured and applied to generate mutations. We need to ensure that once a kind of radiation is applied, it works to establish mutation before the round of replication. So the radiation can generate an effect, or when a radiation generates an effect, it needs to be established to lead to mutagenesis before the round of replication is complete. You need to affect the gametes. So ionizing radiation is actually a bit rarer because it's generated as it's obtained from artificial sources, and its radiation is more dangerous, is more deleterious. It generates free radicals, and ionizing radiation not only generates free radicals, it's more deleterious, and it can directly affect all kinds of gametes and living cells during the ongoing rounds of replication. Even it can penetrate to opaque surfaces, glass surfaces, and hence ionizing radiation is a bit rarer form of mutagen. The most prevalent form of mutagen, which is used in genetics, is non-ionizing radiation called UV radiation. Why? Because non-ionizing radiation generates or leads to certain mutagenic effects which are less deleterious, and hence such effects can be repaired and regulated. Non-ionizing, the effect of UV radiation can be repaired, which generates mutagenesis, and such a mutagenesis can be experimented with UV radiations, carrying out the experiments of mutagenesis, and the effect of UV radiation can be reversed and controlled. So while UV radiation, the important source of UV radiation is the germicidal or UV lamp, which we use for biological experiments. UV radiation can affect surfaces, can affect cells which are exposed or present on the exposed surfaces. And most importantly, a very important issue is that the UV radiation, or even the ionizing form of radiation, is measured in certain units. So if we need to apply these radiations for mutagenesis, we need to have a perfect measure for these radiations. So UV radiations and ionizing radiations are measured in terms of a unit called radiation absorbed dose, that is RAT. Now when applied, in order to apply the radiation for mutagenesis, we need to measure the dose of UV radiation or the dose of ionizing radiation applied to generate mutagenesis, which is expressed in terms of RAT, and 1 RAT, that is radiation absorbed dose, corresponds to 100 ergs per gram. And when a larger amount of dose is applied, we have a still larger unit to measure, that is called Gra. 1 Gra is equivalent to 100 rads and measures large amount of UV radiation and ionizing radiation applied to generate mutagenesis. Now, one very essential feature is the reason behind the fact that UV radiation is the most prevalent form is that not only is does it produce the effects which are less deleterious, not only is it of higher wavelength, readily available, and is it of lesser energy, but also it generates a series of effects which can be reversed and repaired. Once, if we manage to repair the effect of UV radiation before it's stably inherited in course of the round of replication, we can prevent mutagenesis and the effect can be reversed. So the effect of UV is actually reversed by different repair systems called reactivation repair, SOS repair, mismatch repair, with the help of activity of certain enzymes and so on. So before moving on to the way that the effects of UV radiation can be repaired, we need to take a look over the mechanisms of the ionizing and the non-ionizing radiations. So let us again go for a very short break, and I'm coming back with the third part of my lecture, which will be all about the way these ionizing and non-ionizing radiations work, their mechanisms involved in generating mutagenesis, and the way the effect of UV radiation can be reversed, and the reasons behind its more prevalence in experiments of mutagenesis. So till then, now let's take a very short break, and till then keep on browsing through net and keep on going through your books, reading all about mutations. I'll be returning back after taking a very short break.